Good morning, everybody. My name is Soumya Kandi. I am from Abbvie. I work in a department called Quantitative Translational ADME Sciences. We used to previously be known as DMPK, or Drug Metabolism and Pharmacokinetics. So today, I'm going to talk to you about endogenous molecule bioanalysis from Formalin Fixed Paraffin Embedded tissues. Specifically, there's a particular protein called MLKL, and I'll talk to you about it. First, I want to take a moment to thank all the authors who have contributed to this deck, either by adding data directly, intellectually, or creating the infrastructure needed for this research to happen. There are several people from the department I am from. My manager is sitting in the audience today. There are also colleagues from precision medicine, discovery immunology, pathology, who have contributed to this work. And last but not the least, Samir and Dave, who are sitting in the audience as well, who have worked with us every step of the way to make this work happen. There are several people who have contributed in their own different ways to support this research. So I'm going to give a quick biological context as to why we are studying this protein called MLKL. It is called, MLKL stands for mixed linkage kinase domain-like protein. This protein is involved in the necroptotic pathway. It is a key mediator of necroptosis and is associated with ulcerative colitis. So in ulcerative colitis, we see inflammatory cell death as a hallmark. And the request that came our way for custom bioanalysis was to measure MLKL from biopsy colon sections. So this paper very nicely outlines what an FFPE tissue is. We have formalin fixation of the tissues and then paraffin embedding. So we have the scaffold needed for good sectioning. And traditionally, these sections have been available for workflows like histology, immunohistochemistry. Now, in the recent decade or so, these applications have expanded a lot. And this paper does a very nice job showing the different steps that are involved in FFPE tissue analysis. And you can notice that there's a lot of manual processing involved in a non-covarious setup with both the deparaffinization, rehydration steps, where it involves a lot of processes that are integrated from immunohistochemistry, histology workflows. And then there is the heat-induced antigen retrieval, homogenization. Then there is some kind of protease-based digestion, followed by enrichment. And then you can take these through protein bioanalysis. There's also the proteomics aspect or part of the workflow that you could take these samples through. The place that I think covarious fits in very nicely to make this workflow more high throughput are these three steps that I've highlighted. And I won't go into too much detail. Anu did a really nice job showing how it can be made more high throughput. And so within our shop, most of the bioanalytical workflows for proteins follow this general schema where the samples are homogenized. So these are soft tissues or cell pellets. They're taken through homogenization, a sample cleanup either upfront or downstream after trypsin digestion, and then LCMS analysis. On the chromatographic separation side, we have one-dimensional liquid chromatography system. So we have high-pressure liquid chromatography. And then we also have a 2D HPLC system that is set up. So these are hard-cut methods that we typically use for enrichment online. And then on the mass spec side, I don't think I need to explain a lot to this forum here, but I just want to just touch upon, this is a triple quad instrument that we're using, where in Q1 we have selection of the parent ion, fragmentation, and then there is selection of the product ion. So I'll be referring to these parent ion to product ion as transitions that we're monitoring, and then we have detection of these. So we're building out these calibration curves, and there's a lot that goes into consideration for bioanalysis of a protein. So we got to determine the normal ranges within the healthy population and the diseased population, and then also have assessment of accuracy, precision, determination of the surrogate matrix. It is not a trivial task to identify a true null for surrogate matrix for endogenous molecules, because either there's matrix matching, and because this is an endogenous molecule, you would expect a basal amount of this protein to be always present. So on the instrumentation side, we are using a 7500 SciX instrument. It's a triple quad. And here are some of the transitions that I'm showing you at the bottom. On the LK side, as I mentioned, it's a hard cut method. So we have two-dimensional chromatography. So we have the first dimension being a basic mobile phase, whereas second dimension is an acidic mobile phase. We're using a C8 and a C18 column here. And as you'll notice, these are both isocratic methods. So the idea is to have maximal selection for our analyte of interest while depleting out the interfering molecules. So this system was actually set up by Ken Bruder-Voorhees in our lab. If you're interested in learning more about it, I would highly encourage you to check out these references. As a matter of fact, we are trying to reach a GLP-like workflow with our efforts. And so if you're interested in learning more about fit-for-purpose bioanalysis, my manager is giving a talk tomorrow at the DMPK focus group, so I would highly encourage you to attend that as well. So Soumya helped set up this workflow for us, where FFPE tissue is collected in this 96-well plate. These are taken through AFA treatments with the tissue lysis buffer, some antigen retrieval process over here. Then we have another round of AFA treatment followed by sp3 cleanup after reduction alkylation, trypsin digestion, and then LCMS analysis. We're using an LERSE220 for this. So today, I'm going to show you two kinds of data, one that comes from a triple quad instrument, and then another that is from Proteomics dataset. So firstly, we tested two different power settings, a low power setting and a high power setting. And we see that there is detection of signature peptide from endogenous MLKL protein with both settings. There is an increase in the intensity with the high power setting compared to low. And just qualitatively, while we were preparing these samples, both Samira and I, we noticed that there was more complete emulsification of these tissue scrolls compared to the low power setting. And even from the data, the yield that we are seeing an overall increase. And there is also a better precision. It's kind of hard to see on this slide over here, but there is a 12% precision that is associated. I do want to mention that there is a stable isotope labeled internal standard for the peptide that I'm spiking in. And these are, both of these analytes have retention times that coincide. So now that we had tested the sample prep workflow with covariance system, and it worked beautifully with the high power settings, we didn't want to spend more time with the optimization, but rather go into actual biological samples and test out the workflow, see what do we see with proteomics, what are we seeing with targeted protein quant. So the study design is as follows. We have colon tissues from ulcerative colitis donors as well as healthy donors. We have two kinds of FFPE tissues, tissue blocks, resection tissues, and then biopsies. Now, the difference between resection and biopsy, resection tissues are surgically removed parts of colon. Biopsies are non-invasive or less invasive procedures that are performed during a colonoscopic examination. So some proteomics data here. This work was performed by Jan Schabal and Gregory Potts. We are seeing anywhere between 5,000 to 8,000 proteins being detected on the stem stop. And there are several proteins that are upregulated and downregulated in ulcerative colitis in comparison with the healthy tissues. We are seeing that MLKL is buried deep within this volcano plot. It doesn't show any statistical significance. Not that we would expect it. It's a very small sample size. But we are seeing a very nice differentiation on this PCA plot between healthy tissues and ulcerative colitis tissues. Now, just for a quick check, a quick sanity check, if we visualize the network of proteins by the biological processes terms or Go terms, we see that the proteins that are downregulated would be expected to be downregulated. So those that are associated with the integrity of the colon lining are downregulated in ulcerative colitis. And those that are expected to be higher in expression, so those are that are associated with immunity, are upregulated. And ulcerative colitis is an autoimmune disease. So we are seeing that the data is making sense on the proteomic side. From the targeted protein quant, we are seeing that there is MLKL signature peptide detection from all of these samples. So, oops, my apologies. Yep, there we go. So we are seeing MLKL signal to noise ratio over here. If we consider the analytical lower limit of quantitation as 10, we are seeing that all of these are in the quantifiable range. Here's the peptide concentration that has been determined by NanoDrock. Now, when we plot this data, we are seeing that between healthy resection and ulcerative colitis resection tissues, there is an increase or an upregulation of MLKL signature peptide. However, this data trend actually breaks down when we look at biopsy tissues. The reason for this was kind of concerning as to why were we seeing this discrepancy in data sets. As you can see on the right side, I have extracted ion chromatograms from both colon resection as well as colon biopsy. And these were peaks that were well resolved, that were identifiable with a good enough signal-to-noise ratio. And so we went back into the immunohistochemistry data. And here's some semi-qualitative data, quantitative data from both healthy tissues as well as UC tissues. And we are seeing that there is an increase in MLKL staining in ulcerative colitis, and the mucosal layer is primarily where MLKL is being expressed. And that is also much more exaggerated, expanded within ulcerative colitis compared to healthy. So then we went back into the data and decided to have a trained pathologist determine the mucosal area as well as the total tissue area. So when we use mucosal area as the normalization factor, now all of a sudden, this data kind of trends in the direction that we would expect it to. We're seeing that not only does it make sense for the resection tissues still, but for biopsy tissues being as small as they are, it kind of makes sense that when we use the mucosal area where MLKL is being expressed, that the data trends in the right direction. So I want to summarize that this proof of concept testing was very successful, and we are very excited that we were able to get it to work from biopsy tissues, something that we were aiming for within our experiment. This has been a collaborative set of experiments, and I think it expands out the different kinds of questions that we want to ask. And this was a pilot experiment, so I can imagine us wanting to test out multiple different tissue types to increase our N and therefore power our analysis. All of these samples were able to provide us with detectable and quantifiable levels of MLKL. There is some work to be done in terms of wanting to assess the preparation of a calibration curve and things like that. But I just want to point out one last thing, that it is absolutely necessary to determine what the right normalization factor is. Because without this, we were seeing the data trend in opposite directions with biopsy tissues. But when we chose the normalization factor that made sense for this, it helps with determining what is the, having a better informative data presentation, so to speak. With this, I will conclude my talk, and here are some of the references if you want to check out any of the aspects of this work. And I'd be happy to take any questions. Thank you so much for your attention this morning.